Unit 2 is cell biology. Cell biology, there are like 5 components that we need to deal with. One is the cell structure, cellular organelle structure, cell membrane structure, function, and transport. Part of the cell membrane contains this cell membrane structure, function, and cell transport. That's one thing. The second thing is cell organelles. Okay, cell organelles contains, all the different like mitochondria, chloroplast, especially microtubule, from where the questions been mostly asked. They don't generally ask question from very basic part of the cell organelle structure and function, but from microtubule, which is really important, and the junction between the cells, like tight junction, gap junction, desmosomes. That's where they ask questions mostly. And then vesicular transport. Vesicular transport is all about, like, it's not membrane transport but also the transport system inside a cell. Like there are two types of transport while the transport goes through the cytosol. One is without entering into the ER, and another one is entering into the ER, the protein transport mechanisms inside the cell. That's another important thing. So these are the three things that we intend to talk today. And apart from that, the other things that we left up in the second part is the operon, the idea of operon concept, which is also kind of bridge between module 8, you can say, which is bacterial genetics, and this one. So today we'll focus first on the cell membrane structure, function, and its transport. Now actually, from cell membrane, what we need to understand is the structure of the membrane. What are the components of the membrane. I hope all of you can see the whiteboard and the PowerPoint loaded into the whiteboard. So what we intend to know here is the membrane composition. And we all know about the membrane composition very much, because cell biology is a part which all of us at least know. So what we know is the membrane composition. It's composed of lipids, proteins, and carbohydrates. And the whole idea of the cell membrane has been altered from different years. Right now we know it's a fluid mosaic model, and the fluid mosaic model says like the phospholipids out there. As you can see, these are the phospholipids. Phospholipids, they form a bilayer, and proteins are embedded in that phospholipid bilayer. And there are also other components like cholesterol, which are also embedded with the hydrophobic region of the phospholipids. Now this phospholipid single-handedly is the most important part of the cell membrane, because phospholipid contains two separate types of site, hydrophilic head and hydrophobic core inside, hydrophobic tail. Apart from phospholipids, there are other types of lipids that are also found in the membrane, mostly phospholipid, but apart from that, glycosphingolipids and cholesterol, both of them are present. Normally the most of the constituents of the membrane are made up with phospholipids, but there are few regions where we find high density glycosphingolipids, more glycosphingolipids compared with the phospholipids, and more cholesterol embedded in those regions. Now those regions look a little bit different than the rest of the cell membrane. It's something like it's a river, and on the river, on the surface of the water of the river, this few things like the sphingolipids, cholesterol, they make what is known as rafts. River rafts. People raft in the river with something which can float. So things like that, you can identify glycosphingolipids and cholesterols linked together, and they try to be present in few locations of the cell membrane more compared to the other places. So those regions are the places which are taken part in cell signaling process. Okay, we call them lipid rafts, because this type of rafts are made up with lipids, which are cholesterol and glycosphingolipids. So more of a dense region with cholesterol and glycosphingolipids, they make a raft, and those regions are involved with cell signaling, cell contact, and all these different things. Now the question is, this composition of the membrane varies for different types of organism. Okay, not only they vary with different types of organisms, but also this membrane composition may vary with different region of our body. If you take liver cell membrane, the composition there, and if you take the intestinal lining cell and the membrane of that, there will be difference in it. So these things are there. Okay. Now if you look at two organisms, one in the tropical region, another one is in the Arctic region, you'll also see difference in lipid composition in all their cell membrane. Now what you need to understand here is the role of cell membrane in an organism. The role of cell membrane is very, very important. The role is to keep the cell, the cytosol, away from the environment. Because if you remember one thing, that the whole environment and the cell separates the cytosol from the environment with the help of only cell membrane, and that single layer creates a lot of difference inside the cell. That means, you know, outside the cell entropy is always increasing, thermodynamically speaking. Inside the cell is a place where there are organization. Okay, where there are organizations. So something is different about the cell membrane that creates and separates, which is making a closed system, makes the cell as a closed system. Now another very important role of cell membrane is helping the cell in physiological function. Now what are the physiological functions that we want to talk about here? The physiological functions are, you know, phospholipid mobility. Phospholipid mobility helps the cell to perform different functions like interacting with the neighboring cell, functions like help with the cell signaling or respond to a cell signaling, all these things. So for example, if you look at here, we are going to see few of this phospholipid mobilities. There are mainly three types of mobility that we can see. Lateral diffusion. The lateral diffusion is where a phospholipid moves on the same leaflet from one place to the other. Then there are this rotational movement. Rotational movement is the movement of a single phospholipid around its axis. Okay. And the third one is flip-flop movement. Flip-flop movement is when the phospholipid transfer from one leaflet to the other. They can be like from inner leaflet to the outer or from outer to the inner. Now the question is, what are the different types of movement? So the lateral movement, as I told you, so a lipid is moving in that same leaflet. So hydrophilic head is replacing another hydrophilic site, and hydrophobic tail is also providing and replacing another hydrophobic site. So ultimately it's very easy to achieve, right? There is no problem, because hydrophilic moves to hydrophilic, hydrophobic moves to hydrophobic. So it occurs 10 to the power 7 times per second on average. On the other hand, flip-flop movement is very rare. It occurs once a month because it is difficult to achieve. Why it's difficult? Because a lipid needs to move from one leaflet to the other leaflet. So a lipid contains this head region, and this head region needs to be transferred through the hydrophobic tail region of the lipid core, then to reach on the other side. So that requires the help of different proteins as well, you know. And example for such, you know, you can see either that can be spontaneous, which is very rare though, bidirectional spontaneous movement, or sometimes they also need the help of proteins and requirement of energy in terms of ATP. Those proteins are known as flipase and flopase. So there are different examples of that, you know, flipase, flopase, scramblase. These are the example of the helper proteins that they help these phospholipids to move from one leaflet of the cell membrane to the other leaflet. For example, flipase, which is known as a P-type flipase, flips the phospholipid from the outer membrane to the inner membrane. Only single direction, inward movement. While ABC flipase helps them to transfer from inner membrane to the outer membrane. So this is outward movement, which both this case they need ATP. And there is another one which is bidirectional movement, where one move to the inner side, another can go to the outside. So this requires a calcium ion for the proper functioning of that enzyme. The enzyme name is scramblase. They don't need any ATP here as an energy source, but it's like a channel which helps them to guide these phospholipids to move from one leaflet to the other. It can do so by both the way. Okay. So here you can see this picture in separate ways. You can see the flipase, the flopase, and actually the second example that we saw, the ABC flipase, is also known as the flopase. Okay, where they take this phospholipid from inside to the outer side. Okay. The lateral movement is required for, cell signaling process. All the type of cell signaling, if you remember, any basic type of cell signaling, for example, receptor tyrosine kinase, mediated cell signaling, or even 7 transmembrane receptors that are there. So in all this case, they need to interact with their corresponding cytosolic constituent, right? And that thing is only possible. If they move along the cell membrane. Because if I draw it something like this, see, something like this, there is a protein here. So say this is the receptor, and this is where the cytosolic, the enzyme is embedded. So once the signaling molecule binds to the receptor, this signal receptor complex needs to interact with this enzyme. For that, the enzyme needs to be in close contact. So this contact is never possible if there is no lateral movement. Okay. And this flip-flop movement is important to maintain what is known as lipid asymmetry in the membrane. So what is lipid asymmetry in a membrane? Lipid asymmetry is something which says that there are specific lipids or lipid components which tend to be present to either inner leaflet or outer leaflet more. So the percentage of that lipid is more outside, less inside, or more inside, less outside. There is this distribution always. And there are few lipids which are present at the same concentration, both in inner as well as outer leaflet. These two separate sites is known as exoplasmic site, which is outside, and cytosolic site, which is inside. Okay. Now the question is, why this thing is helpful? Why distributing the different lipid molecules in and out in a different concentration will help us? Now the answer to it is that this acts as a signal. This acts as a signal for multiple physiological activities in cell. For example, if a cell is undergoing stress or is infected by an infectious agent, or let's say the cell's normal behavior has been altered and the cell is turned into a cancer cell, to provide a signal to the neighboring cell or to the immune system cells, that cell will flip, will change the lipid asymmetry. For an example, if you look at here, that in this graph you can see one thing clearly. The example like phosphatidylcholine, phosphatidylinositol, phosphatidylserine, right? Sphingomyelin. If you look at the examples, phosphatidylcholine tends to present more to the outer side than inner side. Okay. Same thing for sphingomyelin. But phosphatidylserine present more to the cytosolic side, less to the outside. Phosphatidylethanolamine more towards the cytosolic side, less outside. So now a cell which is normal and behaving normally, they should have more phosphatidylcholine outside and more phosphatidylserine inside. Now if the cell undergoes any stress, what they can do is flip this. So it will flip this asymmetry. So phosphatidylserine outside, that will act as a signal that the cell is undergoing stress and the cell should be killed by introducing the apoptosis pathways. Okay, another example with the same phosphatidylserine. When the phosphatidylserine is reversed from its asymmetry, then it also signals the body to produce the blood clotting cascade in the wounded site. So depending upon the situation, in a wounded site where there is a flipping of this phosphatidylserine outside, that signals the formation of blood clot. On the other hand, for a normal cell cycle is unregulated and the cell division occurring, that's the state. That cell can signal by flipping the serine, and so that the neighboring cell can trigger the apoptosis response. Okay, so this lipid asymmetry or change from this asymmetry acts as a marker for cell death or act as a signal for the blood clot. These are two examples that I said. There are different asymmetry present for all of it. Now for group B, you need to remember few of that. For example, which one presents outside, which one presents inside, the distribution. That you need to know for group B. Now, the membrane fluidity we are talking about, it is also regulated by few facts, few things. You know, what a membrane fluidity depends on. Fluidity depends on, first of all, the length of the fatty acids. Because all the phospholipids, they are made up with fatty acids, right? Two fatty acid chains are there. So the length of the fatty acid is one thing that regulates the fluidity. The type of fatty acids, whether the fatty acids are saturated or unsaturated, that's another important thing. And third is cholesterol. These are the three things that regulates the membrane fluidity. So the first thing, length of the hydrocarbon chain. Shorter the chain, they increase the fluidity. Longer the chain, decrease the fluidity. It's quite logical, because if you imagine long chain, so they have long area, surface area to produce this hydrophobic interaction. Short chain, less interaction, and the hydrophobic interaction is the one that keep them frozen, right? That keep them frozen. That's how the cell membranes can be sticky, because it's lipid actually. The same thing happens in winter time with oil and fat, in winter time and in summer time. In winter time you'll see it's like frozen. There's no much mobility. But in summer time much mobility because of the heat. Okay. So what happens in here, the more they get the length for the fatty acid, the less the fluidity. Okay. Now second thing, regarding the configuration of the fatty acid, saturated or unsaturated. You know, there are two types of this fatty acids out there. Saturated means there is no double bonds that are present, like there is no double bonds or kinks present in this hydrocarbon chain, because a cis double bond, because normally this is how the hydrocarbon chain will look like, but the cis double bond will make the structure bend, and from that part onward there will be what is known as a kink, something like this. Okay. Yes, it's known as a kink. So what we know here, these are saturated, these are unsaturated. So more unsaturated fatty acids will increase the fluidity, because if it's saturated fatty acids, it will have a straight chain like that. So the interaction will be compact. The hydrophobic interaction will be compact. But if they have these kinks, then there will be gap between the hydrophobic core. So the interaction will be less compactness will be there, so more fluidity will be there. Got it? And the third thing is the presence of cholesterol. Now this one acts as a buffer. Presence of cholesterol act as a buffer. Why as a buffer? Actually, cholesterol can make a membrane more fluid, it can make it less fluid, depending on the temperature. So there is not a single answer that if cholesterol is present, it will be fluid, it will be congested, nothing like that. So presence of cholesterol can make it more fluid or less fluid. But cholesterol is present with two separate interactions, because you know these are phospholipids, and phospholipid have the head, hydrophilic head, and the hydrophobic tail. Cholesterol's most of the structures, the ring structures, are hydrophobic, so they are bulkier structure that will be present in contact with the hydrophobic tails, while cholesterol has only very tiny hydrophilic region, that is only a hydroxyl group. So the hydroxyl group that is present will be in contact with the hydrophilic head, where rest of the bulkier region is present in the hydrophobic core. Right? So let's look at the role of cholesterol. See, this all structure, this is hydrophobic because this is hydrocarbon structure. This one is only hydrophilic. So what we can see here is, you can see the cholesterol can hinder the compactness of phospholipids. But how exactly it acts as a buffer? Now at warm temperatures, for example more than 37 degrees Celsius temperature, the body temperature it is, cholesterol restrains the movement of phospholipids. Because you know at 37 degrees Celsius or more, the membrane fluids like the phospholipids start migrating on the different side in the same cell membrane, because it starts to become more fluid. But cholesterol holds on to the neighboring phospholipid so that they become less fluid. Got it? Now at cooler temperature, this phospholipids, if normally phospholipids are present, they will form more compact structure. But presence of cholesterol in the middle hinders the compact structure formation, thus making it more fluid. So in cool temperatures, cholesterol increase fluidity in the membrane. At warm temperatures, cholesterol decrease fluidity in the membrane. Got it? It prevents at cooler temperatures, it allows the lateral movement to occur in a steady rate, or at warmer temperatures it will hinder the movement, because there will be a lot of movement, because at high temperatures this phospholipids continue to move, and that may create pores in the membrane. That thing is prevented by the presence of cholesterol. Because a cell membrane has these two types of state, you know, totally liquid state, another one is a crystalline state, which is semi-solid. At very cold temperature it will be semi-solid, at very warm temperatures it will be liquid. Okay. Now regarding the idea that I told you earlier of the lipid distribution, that cell membranes contain three types of lipids: phospholipids, sphingolipids, glycosphingolipids you can say, and the cholesterol. These are the three major constituents of a cell membrane. Now most of the cell membrane will be made up with the phospholipids, but there are specific regions in cell membrane where we tend to find glycosphingolipids more, cholesterols more. Okay, cholesterol and glycosphingolipid rich regions in the cell membrane, they are known as lipid rafts. So in a definition, what we can say, lipid rafts are small, heterogeneous. Why heterogeneous? Because they carry multiple types of lipids. Highly dynamic, because they alters the structure very fast, the movements are very fast. Sterol and sphingolipid enriched domains that are compartmentalized cellular processes. Cellular process means the process that needs to involve cell membrane in it. Okay, so it's basically cell membrane. Once you look at the cell membrane from top, let's say top view model, if you're looking at cell membrane, you can see this is a place where are dense glycosphingolipids and cholesterol. This is a place, this is a place, this is a place. So in this total cell and cell membrane, 4 regions you'll find, for example here, they carry more of a glycosphingolipids and cholesterol rich part. Those regions will be termed as lipid rafts, and they will participate in the process of cell signaling, cell-cell interaction. Okay, and we know cell signaling, what is the importance of cell signaling and cell-cell interaction. Okay, so it is nothing but a modified region of the cell membrane. One such example of lipid raft region for a cell-cell interaction is in immune system. In our immune system, what we have is this antigen presenting cells like APCs, all the APCs out there, you know, dendritic cells or B cells. So antigen presenting cells, they present antigen to the surrounding what? T cells. So there will be interaction between the T cell and the antigen presenting cell. For example, macrophage showcasing part of an infective agent to the T cell, and T cell will recognize, and there will be an interaction between macrophage and T cell. That interaction requires multiple protein lipid connections. So that needs to be highly dynamic, because the regions which is involved in cell signaling or cell-cell interaction needs to be highly dynamic. Dynamic means the composition of the lipid needs to be continuously changing and moving so that the process can be done fast. That's what working here in the lipid rafts. Okay. Now we've been talking about only one side of the lipids, one side of the cell membrane, that are lipids. But cell also carry another thing. There are proteins. Proteins, like membrane proteins, are the second component of cell membranes. We know the role of lipids. We know why lipids are there. Without the lipids, without their lateral movement, it's not possible for any cell signaling or cell-cell interaction. But membrane proteins are also required for cell signaling. Membrane proteins are also required for providing information to the inside of the cell from getting a response from outside, right? So these connections are really important, because you know, not all the type of signaling, the signaling molecule goes inside the cell. Most of the cases, signaling molecule only interacts to one receptor outside the cell, and that will convey the message inside the cell. Okay, that's an idea, and that's how the whole cell signaling works. And for that, we need these proteins which are embedded to the membrane, right? So the proteins embedded to the membrane here, in this case as a receptor. Okay. Now, example of membrane proteins: integral proteins, peripheral proteins. Integral proteins, there are different types. Okay. what you need to know are the different types of membrane proteins and how to predict their structure, how to know what kind of structure they may have. And second thing is one example for each of this type of proteins. That's what you need to know, okay? And their function. So integral protein means the protein is embedded into the membrane, and peripheral protein is something which is present outside the membrane, okay? So you can see in integral protein there are single pass protein. Single pass means the protein which will traverse the membrane once, and this protein is made up with alpha helix. Most of the protein is components of the alpha helix, okay? Then multi-pass membrane protein. As you can see, this protein is transferring the membrane multiple times, forming what is known as a helical bundle. Also with alpha helix, multiple alpha helix together, that's why we call it helical bundle. So generally these single pass proteins are involved in recognition or as receptors, while these multi-pass proteins also act as enzymes for cell signaling. One such example, GPCR. You know the full form of it? G protein coupled receptor, which helps in a G protein mediated signaling process, which is a key to the signaling of most of the cell cycle mediated signaling or cell division mediated signaling with the help of growth factors. And this is another one which is also multi-pass but unique. Unique means it forms a barrel, like a channel. Like a channel. So if you look at a top view, it looks something like this, like a cylinder. Things can go inside or outside with it. So they act as a channel proteins. So the main job of these channel proteins are to act as transporters. Okay, so these are the three types. Apart from that, peripheral proteins tend to present only one leaflet, one side only. Now these proteins are also anchored, you know, they are sometimes anchored. Anchored means a protein is not transferred or it's not embedded in and out of that whole cell membrane. Instead, that protein is embedded with one leaflet of the cell membrane. For an example here, this is towards the cytosolic leaflet, this is outside the cytosolic exoplasmic leaflet. Because you know there are proteins need to be stick out, outside or inside depending upon the situation. And all these proteins, they act as either enzymes or carriers, you know, carriers, which are another type of transporters. Now when I say transporters, there are two types of proteins as you can see, for integral, another for peripheral. Channel proteins and carrier proteins. Now there is a difference between this channel proteins and carrier proteins. What is the difference between the two? Because we need to understand that clearly to understand the process of membrane transport. Channel proteins create channels or way. For example, this one, channel protein creates door through which molecules can go in and out. While carrier proteins present at one side of the cell membrane. So they alter their structure. For example, channel protein opens up at one side, signaling molecule binds, or any other, not signaling molecule, but any, for example, cargo protein or cargo molecule binds there. And then this channel protein alters its structure in a way that it will open up towards the cytosolic leaflet. So it can take that molecule inside. So both of them helps in transferring a molecule inside the cell or outside the cell. But the approach is different. In case of channel proteins, it's directly through the passage. In case of carrier itself changes its structure to do so. Okay, that's the difference. So here you can see multiple types of proteins as a list: transmembrane spanning proteins, single layer linked proteins, lipid linked type of proteins. Lipid linked type of proteins are where the protein will be in contact with the lipid, with the fatty acid chains. As you can see, they are known as a lipid anchored protein. And there are also protein attachment type protein. Protein attachment type protein is those proteins which are in contact to the integral proteins, okay, with the help of protein-protein interaction, surface interaction. They are not anchored to the membrane, okay. So they are mostly cell signaling associated proteins, okay. Now the question is, all these proteins that we're looking, most of the single pass or multi-pass proteins that we saw earlier, they made up with alpha helix. Single pass alpha helix, multi-pass helical bundle that is present. But channel proteins most of the times made up with beta sheet, because we know there are these only two types of structures that are possible, the secondary structures of proteins that are there to make all the 3D structures of protein, and those structures are alpha helix and beta sheet. So these channel proteins most of the times are made up with beta sheet. So beta sheets are folded, they form what is known as a barrel called beta barrel. Why it's called barrel? Because it looks like barrel. Barrels had a shape something like this. So they form a cylindrical barrel shaped structure. That's why we call them beta barrels. So beta barrels produce these channel proteins. And the unique feature about this beta barrel is that the center region of the beta barrel are hydrophilic, so that any signaling molecule or any other molecule trying to go inside the cell can pass and go inside, because molecules are hydrophilic here. While the surrounding portions are hydrophobic outside, so that the barrel can interact to the hydrophobic core of the cell membrane while embedded to the membrane. Got it? So that gives you another very interesting fact to know regarding the membrane proteins, is what kind of amino acid these membrane proteins are made up with, because there are hydrophobic amino acids, there are hydrophilic amino acids. So what kind of amino acids they are made up with? Now to understand that, we can look at the whole length structure of the primary sequence of those proteins or peptides. So primary sequence of proteins, if we check, there is always a list of amino acids linked with one another. Now among those list of amino acids or linked amino acid structure that we can see, we need to find out how many of those amino acids are hydrophobic, how many of them are hydrophilic, okay. So if I look at here, this one example, we are trying to look at a stretch of amino acid sequence, a protein sequence, the primary data sequence of a protein. Now in this sequence, in this graph, what we can see, 0 is the standard range, and above that we add plus, and then minus means below that particular range. Plus here means that region will form hydrophobic, and minus hydrophilic. And in the bottom we start writing the number of amino acids. So from 0, 1, 2, 3, 4, 5, 6, and you continue like that. So we are going to look at a stretch. We are going to look at a stretch of amino acid sequences there. So from 0 to 50, or say 60 here, that one from 0 to 50 or 60. If I use pointer, I couldn't draw. That's a big problem of using pointer. So you can see the pointer on the recorded class, because I'm actually using the pointer, you can see, but probably in the live class you may not see. So here 0 to 50 is the place where the stretch of amino acids are hydrophilic in nature. This one, hydrophilic in nature here. Then again from approximately 60 to 90, that range hydrophobic. So one hydrophobic domain, then another hydrophilic domain. So in this protein structure we have a hydrophilic domain at the beginning, this one, then a hydrophobic domain, then again a hydrophilic domain. So once we look at the structure of the protein, it looks something like a single membrane pass protein, because a single membrane pass protein should carry one hydrophobic site. Remember? Isn't it? Because it looks something like this is the cell membrane and this is the protein. So this region will be hydrophobic, this will be hydrophilic, this will be hydrophobic. The same type of structure that we can see here, Example, glycophorin, single pass membrane protein. The second type, bacteriorhodopsin. Here we can see that we start with again N terminal and then to the C terminal. We continue with a stretch of amino acid sequence. Okay, so the first stretch of the sequence here, hydrophobic, second one hydrophilic, then the third one again hydrophobic, hydrophilic, hydrophobic, hydrophilic, hydrophobic. It continues to do the same approach of hydrophilic and hydrophobic, hydrophilic and hydrophobic. So there are total 7 stretch of hydrophobic sites. That means this protein passing the membrane 7 times, right? So mostly this type of proteins are 7 transmembrane proteins because they transverse the membrane 7 times. So you can see the difference between both this type. This is the primary structure of this protein, which is only single pass, but this one the 7 pass membrane protein. Now this type of graph, which is also known as a plot, helps us to find out the structure of a protein that is present in cell membrane, and we call it hydropathy plot or hydrophobicity plot or hydrophilicity plot. Anything, same name, same thing. Hydrophobicity plot, hydrophobicity plot, or hydropathy plot. So hydropathy plot is a quantitative analysis of the degree of hydrophobicity of an amino acid of a protein, which helps us to predict the structure of protein of the cell membrane. Okay. The dark shades of green represents hydrophobic regions. Functions of membrane proteins. So apart from the structure, because you know the structure is always associated with the function of proteins. So if a protein structure is barrel shaped, that protein is too involved with the transport. If it is anchored or if it's linked to another protein on the surface of the cell, it is probably linked with the cell signaling. So we can predict the function based on the structure. Okay, so there are multiple functions played by the membrane proteins, mainly transport, enzyme activity, signal transduction, cell-cell recognition, intercellular joining, and attachment to the cytoskeleton and extracellular matrix region as well. So all these functions can be represented with all these pictures. As you can see, the proteins associated with transport, there are numerous number of transporters that are available, you know, which can act without the energy utilization, with passive transport, and with the energy utilization known as active transport. Then the enzyme can act as an enzyme activity, like the protein can act as an enzyme, that may also occur. And the examples of enzymes, you know, the G protein coupled receptors, they are also involved with the same process. They can also work as a signal transduction molecules, okay, because they can act as a receptor and cell signaling or primary cell amplifying molecule in the membrane, which provides a signal to any of the components inside the cytosol. Cell-cell recognition, because you know these proteins are sometimes also linked with carbohydrate moieties on the surface. That carbohydrate moiety with the proteins attached to the surface forms extracellular matrix. This extracellular matrix regions helps the cell to anchor with the tissue lining or to interact with the neighboring cells to make a compact tissue structure, which is very much important while the cell was dividing to make the whole organism during the development of any organism, as well as the cell-cell communication, you know, cell-cell contact, as we see in immunology, immune systems, intracellular joinings or cell-cell interactions. That's another important thing. Another thing of this proteins, that they will also contact themselves with the cytoskeleton of the cell. So because you know if you look at the cell membrane and the proteins are embedded to the membrane, and cytoskeleton holds the structure of the cell together, because cytoskeleton will provide the shape of the cell. Otherwise the cell is nothing like a jelly like structure, because the cytosol is also liquid, most of it is made up with water. So who keeps the structure as like round or any other structures that the cell have is with cytoskeleton. So these proteins also interact with the cytoskeleton to make that structure component steady. As you can see here, this is actin filament drawn here, and you can see the protein is interacting with the actin filament inside the cytosol to keep the structure of the cell intact. On the other hand, that protein also interacts to the extracellular matrix, and the extracellular matrix is mostly made up with all these carbohydrate moieties coming out from the different protein surface of the cell membrane. Okay, so all these functions are played by cell membrane, but among all these functions we'll study the whole cell signaling process as cell signal transduction process in unit 4, and today we are going to talk about two things. One is the transport, and the second one is the cell joining or intercellular joining, making of structures where the cell and cell interact with each other, forming tight junctions which act kind of a stitching material between the cells, because in multicellular organism the cells always tend to stick to each other. They start to stitched to each other, and that forms what is known as a tissue lining. Okay, so we need to understand that intercellular junction of the cell, as well as we'll talk about the transport. So before understanding the transport, we need to know two fundamental ideas. We need to make it very clear, and I hope all of us know these two ideas. These are fairly simple idea. Diffusion and osmosis. These are the two phenomena that we want to understand, we need to understand, because anything that goes in and out of a cell either follows this diffusion or follows osmosis, the rule of osmosis or diffusion. Otherwise nothing can go in or out. So first of all is, what is passive diffusion, or simply we can call it as a diffusion. Diffusion is a process where solute from higher concentration move to the place of lower concentration of that same solute. So it will continue to move from high concentration to low concentration until it reaches an equilibrium. The equilibrium means both the sides will carry same concentration of that solute. That's passive diffusion, you know. And anything can go inside the cell that is permeable to the cell membrane following this passive diffusion law. Anything can go inside until they find all the rules that a cell applies for the transport of molecules, which we are going to see in a moment. So molecules like oxygen, any gaseous molecules like oxygen, carbon dioxide, it can easily go inside or come out based on the concentration gradient with passive diffusion. They don't need any accessory transporter, they don't need anything else. They can directly go inside and come outside just based on their concentration gradient across the membrane.